Hi, today I'm going to discuss the translational process, which is the last part of central dogma. Earlier, I have discussed replication, transcription, and now this is translation. So, in replication, you haven't seen the copying of DNA from DNA, and then transcription, synthesis of mRNA or formation of mRNA from DNA. And in translation, you will be converting the mRNAs to the amino acids or polypeptides and then the proteins. So converting genetic information into proteins called translation. mRNA is code for proteins which are synthesized at ribosomes. So the proteins are synthesized at ribosomes. And here the sequences are representing the DNA, which is double-stranded ATGC strand. And this is the RNA sequence, which you can see as a single strand. And then this single strand mRNA is converted to protein. So this is from transcription to translation, you can clearly see here. And now, if you also might have seen somewhere or heard the codons. So this is the table which you can easily remember or understand. The table of three bases. Like here you can see UUU, UUC, UUA, UUG. So these are basically coding for different amino acids. And these three, for example, one direction you can see the base U and then C, A, and G. Here again, U, C, A, and G, and then here U, C, A, and G. So you basically mix them up, and their combinations are 64 combinations. And out of 64 combinations, you can see that 61 combinations are coding for the amino acids, and out of which 60 are normal amino acids, while one AUG is considered as a start codon, which codes for methionine, and then the three codons are for stop codon, that means a stop signal. So if you could understand and memorize this table, it will be really worth for you to work in the field or study the biology field. Now, how this translation process could work. So you need to understand in detail the protein synthesis, which is like the final product protein as a result of RNA translation. So in one step, you can see here an enzyme called amino acyl transfer RNA synthetase attaches amino acids to their corresponding transfer RNA molecules using energy from ATP. Each amino acid has its own transfer RNA molecule with the anti-codon for that amino acid. And then you could see a series of process like a small ribosomal unit attaches itself to the 5' end of the mRNA strand, and then it moves along mRNA unit until it finds a start codon. There, the first transfer RNA and the large ribosomal unit joins it. And then the next step, basically, there are series of steps which are required for final protein synthesis. And depending on the your educational standard, for example, if you are in +2 or graduation or some higher level, you need to understand the process of translation or protein synthesis. In general, this protein synthesis process is in three steps, but here you can see transcription DNA to RNA, then RNA processing, remove introns, apply 5' cap, apply poly A export mRNA from the nucleus, and then mRNA is translated into polypeptide by the ribosomes, and these polypeptide may be delivered to a specific cell compartment or the sites. Polypeptide may require assistance from the chaperone proteins to fold, and then to produce the active protein. Polypeptide may associated with other polypeptides, ligands, effectors, coenzymes, or cofactors. Now, protein synthesis flow chart can be summarized. For example, DNA to transcripted, like transcription pre-mRNA, that is here post-transcriptional modification could also take place. Then finally mRNA is formed, and then the mRNA is translated into proteins, and then post-translational modification is possible in the newly formed proteins. And this is like here, you can see proteolysis and protein foldings. These are the overall conclusive remark which you need to understand in the part as a part of central dogma. Now, the three steps involved in translation are initiation. The ribosome assembles around the target mRNA. The first transfer RNA is attached at the start codon. Then elongation. The last transfer RNA validated by a small ribosomal unit, that is called accommodation, transfers the amino acid. It carries to the large ribosomal unit, which binds it to one of the preceding emitted transfer RNA, that is peptidization. The ribosome then moves to the next mRNA codon to continue the process of translocation, and that creates an amino acid chain. And finally, the last part is termination, when a stop codon is reached, and the stop codons are three, which you have seen it. The ribosome releases the polypeptide. The ribosomal complex remains intact and moves on to the next mRNA to be translated. Now, the complete map of these three steps, initiation, elongation, and termination, could be seen here. So from here, you can see the initiation process takes place. During initiation, the components of the translational apparatus come together with an mRNA, and the transfer RNA carrying the first amino acid binds to the start codon, that is AUG. And then during elongation, in the second step, amino acids are brought to the mRNA by transfer RNAs and are added one by one to a growing polypeptide chain. And after elongation, the termination step comes into effect. During termination, a stop codon in the mRNA is recognized by a protein release factor, and the translational apparatus comes apart, releasing a completed polypeptide. And that is what you can see here. And then this is recycling process going on. Now, there are some other things also during translation. There are some errors, which I will be discussing, and it has a great importance in biology and biological diseases. Now, in biology, translation is the process in living cells in which proteins are formed using RNA molecules as templates. The generated protein is a sequence of amino acids. The sequence is determined by the sequence of nucleotides in the transfer in the RNA. The nucleotides are considered three at a time. Each such triplet results in the addition of one specific amino acid to the protein being generated. The matching from nucleotide triple to the amino acid is called genetic code. The translation is performed by a large complex of functional RNA and proteins called ribosomes. The entire process is called gene expression. In translation, messenger RNA or mRNA is decoded in a ribosome outside the nucleus to produce the specific amino acid chain or polypeptide. The polypeptide later folds into an active protein and performs its functions in the cell. The polypeptide can also start folding During protein synthesis, the ribosome facilitates decoding by inducing the binding of complementary transfer tRNA anti-codon sequences to the mRNA codons. The transfer RNAs carries a specific amino acids that are chained together into a polypeptide as the mRNA passes through and is read by the ribosome. Now, the basic mechanism, the basic process of protein production is the addition of one amino acid at a time to the end of a protein. The operation is performed by a ribosome. A ribosome is made up of two subunits, a small subunit and a large subunit. These two units come together before the translation of mRNA into a protein to provide a location for translation to be carried out and a polypeptide to be produced. The choice of amino acid type to add is determined by a messenger RNA molecule. Each amino acid added is matched to a 3 nucleotide subsequence of the mRNA. For each such triplet possible, the corresponding amino acid is accepted. The successive amino acids added to the chain are matched to successive nucleotide triplets in the mRNA. In this way, the sequence of nucleotides in the template mRNA chain determines the sequence of amino acids for the specific protein. The addition of an amino acid occurs at the C terminus of the peptide. Thus, translation is said to be amino to carboxyl directed. Now, the mRNA carries genetic information encoded as a ribonucleotide sequence from the chromosomes to the ribosomes. The nucleotides are read by translational machinery in a sequence of nucleotide triplets called codons. Each of these triplets codes for a specific amino acid. The ribosome molecules translate this code to a specific sequence of amino acids. The ribosome is a multi-subunit structure containing ribosomal RNA. And proteins, it is the factory where amino acids are assembled into proteins. Transfer RNAs are small non-coding RNAs, which are 74 to 93 nucleotides that transport amino acids to ribosome. The repertoire of transfer RNA genes varies widely between species, with some bacteria having between 20 to 30 genes, while complex eukaryotes could have thousands. Transfer RNAs have a site for amino acid attachment and a site called an anticodon. The anticodon is an RNA triplet complementary to the mRNA triplet that codes for the cargo amino acid. Now, the initiation and termination of translation. These things we have discussed as from the picture in the previous slides. The initiation involves a small subunit of ribosome binding to the 5' end of mRNA with the help of initiation factors. In bacteria and a minority of archaea, initiation of protein synthesis involves the recognition of purine-rich initiation sequence on the mRNA called Shine Dal-Garno sequence. And then there are some steps which you need to understand in the protein synthesis or the translation of mRNA to protein. These steps require a specific kind of simplified knowledge. And then you also need to understand, if you are interested deeply in translational process, then how the effect or drugs binding affinity, drug activity could be affected at this level also. Then the same process of initiation, elongation, and termination could be seen here in details. Like here, the initiation process you can see there, and then elongation process also you can see how one amino acid is combined with another and another with another, and then thus it forms the polypeptide chain in order to make it the primary structure of amino acid, and then it goes to the protein folding and misfolding. Then errors in translation process. Of course, during such a complex process, biological process, there is a possibility of errors, and this makes biological meaning or meaning in the sense of clinical perspective human diseases. Even though The ribosomes are usually considered as accurate and processive machines. The translation process is just subject to errors that can lead either to the synthesis of erroneous proteins or to the premature abandonment of translation, either because a transfer RNA couples to a wrong codon or because a transfer RNA is coupled to the wrong amino acid. The rate of error in synthesizing proteins have been estimated to be between 1 in every 10 to the power 5 and 1 in every 10 to the power 3 in misincorporated amino acids. Depending on the experimental conditions, the rate of premature translation abandonment instead has been estimated to be of the order of magnitude of 10 to the minus 4 events per translated codon. So the translation errors could be like amino acid misincorporation, transfer RNA misacylation, and then premature termination, read through problem, frame shift issues, splicing errors where exon skipping, failure to remove introns, transcription errors, nucleotide misincorporation, polymerase slippage, folding errors, kinetic trapping, spontaneous unfolding, disruption of folding due to erroneous polypeptide, errors at in post-translational modifications like incorrect proteolytic cleavage, erroneous ubiquitination or glycosylation, and phosphorylation. Other errors in post-translational modification or functionalization is also possible. So this figure I have adapted from Nature Reviews Genetics. Now, the protein cycle, if you want to understand, like the synthesis process, co-translational process, functional lifetime, degradation. So in synthesis, deregulation is stoichiometry of translation components, sequestration of mRNA in stress granules, decreased overall translation rate, then co-translational processes, misfolding due to chaperone depletion, reduced chaperone activity, or low ATP levels. Mislocalization of proteins into endoplasmic reticulum and mitochondria, then functional lifetime, increased protein lifetime, accumulation of damaged proteins during asymmetric division, misfolding dysfunction and aggregation due to damaging protein modifications, reduced repair of oxidized proteins, and dysregulation of chaperone system. Now, in deregulation, deregulation, impaired degradation, reduced acidity of autophagy organelles, autophagy induction and autophagosome fusion, autophagy and proteasome crosstalk, ubiquitination, and proteasome misassembly. So these are the things which you need to understand for biological or human disease problems related things if you are working in this area. Now, regulation, the process of translation is highly regulated in both pro and eukaryotic organisms. Regulation of translation can impact the global rate of protein synthesis, which is closely coupled to the metabolic and proliferative state of a cell. To study this process, scientists have used a wide variety of methods such as structural biology, analytical chemistry, imaging of reporter mRNA translation, and then next generation sequencing based method, that is NGS. Other methods such as to a parenting assay can be also be used to determine the location of ribosomes of a particular mRNA in vitro and footprints of other proteins regulating translation. Now, protein or RNA binding near the ribosome binding site negatively regulates the bacterial translation initiation. This is regulation of translation. Now, there are some other examples of translation regulation. For example, high iron concentration IRE binding proteins cannot bind to IRE, so translation of ferritin mRNA proceeds. And then you need to understand the in particular ribosome profiling, which is a powerful method enables researcher to take the snapshot of all the proteins being translated at a given time period. showing which parts of the mRNA are being translated into proteins by ribosomes. This method is useful because it looks at all the mRNAs instead of using reporters that would typically look at one specific mRNA at one time. Ribosome profiling provides valuable insight into translation dynamics, revealing the complex interplay between gene sequence, mRNA structure, and translation regulation. For example, research utilizing this method has revealed that genetic differences and their subsequent expression as mRNAs can also impact translation rate in an RNA-specific manner. Expanding on this concept, a more recent development is single-cell ribosome profiling, which is a technique that allows us to study the translation process at a resolution of individual cells. This is particularly significant as cells, even those of the same type, can exhibit considerable variability in their protein synthesis. And single cell ribosome profiling has the potential to shed light on the heterogeneous nature of cells and leading to a more nuanced understanding of how translation regulation can impact cell behavior, metabolic state, and responsiveness to various stimuli or conditions. Now, it comes to clinical significance of translational process. As I mentioned that if there is any kind of error at the translational level, it could lead potential disaster or potential human diseases. Translational control is crucial for the development and survival of cancer. Cancer cells must frequently regulate the translation phase of gene expression, though it is not only fully understood why translation is targeted over steps like transcription. While cancer cells often have genetically altered translation factors, it is much more common for cancer cells to modify the levels of existing translation factors. Several major oncogenic signaling pathways, such as MAP kinase pathways, AKT pathways, MYC pathway, Wnt, catenin, SIC pathways, ultimately reprogram the genome via translation. Cancer cells also control the translation to adapt to cellular stress. During stress, the cell translates mRNAs that can mitigate the stress and promote survival. An example of this is the expression of AMPK in various cancers. Its activation triggers a cascade that can ultimately allow the cancer to escape apoptosis triggered by nutrition deprivation. Future cancer therapies may involve disrupting the translation machinery of the cell to counter the downstream effects of cancer. And that is what you need to understand if you are deeply interested in the translational process. So translation process is not just to understand the synthesis of protein from mRNA, but you can also understand in the perspective of human diseases and biological functions. So there, as you know, that the translation process includes three steps: initiation, elongation, and termination. For initiation, you need AUG, amino the codon, and that is methionine, and then UAA as the three codons which are responsible for termination, or known as a termination codon or stop codon. And then in between it is elongation process. And after that, protein translation normally, where you can look for fibroblast, stem cell, synaptic neuron, myocytes, and spheroidal cells. Then if you want to understand the deregulation, then deregulation examples are, the best example is cancer, neurodegenerative diseases, where you can understand the impact of protein misfolding, and then infectious diseases, cardiovascular diseases. These are the broad categories of human diseases where protein translation has potential and crucial roles. Now, if you want to understand the functions of proteins, there are a wide range of protein functions. So, enzymes help to carry out all the chemical reactions performed by a cell and thus our body. And all enzymes are protein. Integral membrane proteins are responsible for the transport of compounds in and out the cell. Then antibodies or immunoglobulins protect us from bacterial and viral infection. Structural proteins such as actin and myosin provide the strength and the structure to the cells and our body. Signaling compounds like cytokines and hormones are essential for the cell to cell communication. And thus, the another most important thing, as I mentioned in the beginning of today's lecture, that you need to understand the codon map or chart. So these codon maps, if you understand as clarity, you will be able to easily understand the realistic of molecular biology. And then this is like a possible, like the genetic code is possible to translate either by hand, you can do it by hand, or by computer. This allows biologists and chemists to draw out the chemical structure of encoded protein on the paper. Now first convert each template DNA base to its RNA complement, as you can see here, and then the template strand of DNA is one of the RNA is polymerized against, and the other DNA strand would be the same as the RNA, but the thymine instead of uracil. So here you can see this this one thymine, and then this is basically replacing the uracil. So now then they split the RNA into triplets and then go for the primary structure of the protein. And after that, you need to understand the protein folding like 3D structure, tertiary structure, and then further protein PTM, post-translational modifications. And finally, there are many computer programs available which could help you to convert all these translational process easily, or even replication, transcription, and translation. Thank you very much for today. So in the end, you need to understand with full clarity, that is translation tables. And thank you very much for better understanding. And also look for some computational softwares or tools which are easily available as a web server. You need to understand and run some of the sample softwares. Thank you very much, and see you in the next class, please.